Hello, my dear students. Today we are here to discuss one of the very important application of biotechnology, which we are calling as enzyme biotechnology. Now, before we start, let us quickly recapitulate in a nutshell what we have studied in the previous class. In the previous class, we have discussed mainly what do we mean by biotechnology, what does term biotechnology basically means. Then we discussed about what are the various areas or domain where biotechnology is commonly applied, or what are the various fields which are benefited by the application of biotechnology. Just to brush up your memory, that was starting from its application in agricultural field with the major breakthrough of producing biotechnology cotton. We produced various organic farming. Then the application went on to the production of various antibiotics. Then we had seen the production of various vaccines to protect individuals against various diseases. It may be a bacterial disease like tuberculosis, or it may be a viral disease like pulse, polio, and so on. The latest application of biotechnology was the invention of vaccine to protect individuals from coronavirus. Then we discuss about what was the origin, how it started. I have narrated in a nutshell about the history of biotechnology originating way back in China in 1700 BC, and that origin which started with the formation of beer by the process of fermentation. Even today, the science is gradually advancing. We definitely have seen a number of major breakthrough like development of monoclonal antibodies, use of gene therapy, application of fermentation technology, so that a large number of compounds are being produced, and they are very effectively utilized nowadays. So, with this enormous growth, a number of pharmaceutical companies, a number of biotechnology companies, they are taking advantages of these improvements, and they are growing just like anything. The entire scenario was discussed in the previous class of mine. Now, today we will be mainly concentrating ourselves with the first application of biotechnology, which we are calling as enzyme biotechnology. Now, in our class today, we will be discussing about what is enzyme biotechnology, what are its application, and what are the various recent advancements. These are the three main areas where I will try to focus on in my class of today. Question is enzyme biotechnology. That is, if we just look at this in a blind eye, you will see application of biotechnology with the help of microorganism to produce enzymes. Application of biotechnology using the microorganisms to produce enzymes. This is the crux or synopsis or abstract of enzyme biotechnology. I will discuss with you about what are the various enzymes which are produced by the help of the biotechnology tools. It ranges from lipase, amylase, asparaginase. We can produce various antibiotics even, amino acids even with the help of this biotechnology application of fermentation. But before I go further, we should first realize why microbes are so much important to produce or generate industrial product. The first thing, they are associated with the ability to attack, degrade and utilize the components of the media and to accumulate them into a fermentation product. The concept goes like this: the microorganism, they are having their own enzyme systems and they are utilizing these enzymes to produce the microbes. They are utilizing this substrate, that is the media ingredients, very nicely, and they are producing various enzymes of economic high value. These enzymes can be an economic product like invertase, amylase and protease, which are having a very wide application nowadays. This slide basically highlights the commercially important products which are used nowadays obtained from microbial origin. Just to see, it started with antibiotics. Various antibiotics like penicillin, a legendary compound which is produced by Penicillium notatum. Next, we add streptomycin in our pipeline, obtained from Streptomyces griseus. Then we have tetracycline, we have erythromycin, we have polymyxin, we have bacitracin. So, a wide range of antibiotics, they are produced with the help of microorganism by fermentation. But remember, microbes not only restrict itself to only production of drugs, they have a very important application in production of solvents. Solvents like acetone, ethanol, butanol, amyl alcohol, they are very easily produced with the help of microbes. Microbes have a very important role to play in production of beverages like wine, like beer, etc. Various food substances like cheese, fermented milk, pickle, soya sauce, yeast, bread, vinegar, mushroom and citric acid, they are effectively produced commercially by the microorganisms. The other products include the vitamins B12, riboflavin, vitamin A, growth stimulants like gibberellins, various enzymes like amylase, protease, pectinase, invertase, they are produced with the help of fermentation. Organic acid like lactic acid, acetic acid, citric acid, gluconic acid, butyric acid, fumaric acid, just to see the range of compounds which are produced from the microbial origin. Microbes, they are also successful in producing various amino acids like L-glutamic acid, L-lysine, etc. They are successful to produce steroids. Particularly, you will be happy to know that there are certain situations where your chemicals, they are not successful in inserting certain functional groups in steroid. But whenever we are using a microorganism, that hydroxyl group they can easily be incorporated. That is, microbes are successful in incorporating, in inserting the hydroxyl group within the steroid moiety very easily, which a ordinary chemical cannot do. That we are calling as microbial biotransformation of steroid. That we are calling as microbial biotransformation of steroids. Microbes have been used in baker's yeast as well as various feed yeast also. Question is, what is an enzyme? Enzymes are proteins that acts as a biological catalyst, which we are calling as the biocatalyst, and accelerates various chemical reactions. The enzyme speed up the rate at which a chemical reaction takes place. It accelerates the rate of chemical reactions. The enzyme generally is not destroyed during the reaction and hence can be recycled and reused time and again. This is basically the structure of an enzyme, and these are the active sites where the substrate tries to get attached. This slide shows the various classes of enzymes and the various reactions they carry out on the various important subclasses. We have enzymes, various classifications like oxidoreductase, like transferase, hydrolase, lysase, isomerase and ligase, and they are the various reactions which are conducted by these chemicals, I mean these enzymes. Now, enzyme biotechnology encompasses modification of an enzymatic structure or catalytic function to yield a novel metabolite or to take part in a new chemical reaction. This technology enables the commercial synthesis of various enzymes with diverse application in the field of pharmaceutical biotechnology, in food processing, preservation, and enzyme. Now, see, when an enzyme is produced, it sometimes so happen that the enzymes they may not be very much stable. The enzymes may not be very much stable. So, to impart stability, sometimes we adopt the process of enzyme immobilization. There are various techniques which we will be discussing in the subsequent classes, but let me just define what is enzyme immobilization. Enzyme immobilization is defined as the imprisonment of an enzyme or a cell on a distinct support or a matrix. We want to protect the enzyme, we try to encapsulate the enzyme within a particular matrix or a support. This support or a matrix on which the enzymes are immobilized allows the exchange of the medium containing the substrate or the effector or the inhibitor molecule. They do not hamper the process of the reaction, but they try to protect the particular chemicals for particular enzymes from undergoing degradation. The practice of immobilization of cell is very old, and first immobilized enzyme was aminoacylase of Aspergillus oryza for the production of amino acids in Japan. Now, this slide shows the various application of enzyme in industry, used in protein, hydrolysis, in food and beverage industry commonly utilized. We applied in starch and fuel, pulp and paper, leather tanning, etc. Now, if we talk of environment, we had used this enzyme for detection of toxic pollutants as well as degradation of waste product. Next, in the field of agriculture, we have applied it in agro processing, animal feed additives, agrochemicals and animal feed from agricultural waste. These are the various application of the industrial enzymes. The other examples include renin for coagulation of milk to make cheese, invertase from yeast and lactase in food industries, cellulase and amylase to remove the waxes, oils and starch coating on fabrics and on to improve the look of the final product. Amylase protease are applied for baking, lipase applied on fruit juice to break down the cell wall of the microbes, protease, lipase, amylase, they are also used, carbohydrate used to convert starch into corn syrup and so on. Now, we are moving on to the most important area. We have seen that these enzymes, they are having a very important commercial activity. That is, the enzymes which we are talking of, they are having a very high commercial value. Keeping in mind of the commercial value, let us see how you are going to produce these enzymes in your laboratory, or you are going to produce this enzyme in various industries. Now, as the source of these enzymes, we have to make two things. One, we have to make the media ready, that is the media on which the microorganism will grow, we need to make it ready, and at the same time we have to make the organism ready, that is the microbes which will be producing that enzyme, that needs to be made ready. If you see in your classes of microbiology in B.Pharm 3rd semester, you have seen that the microbes, basically there are 4 phases, one lag phase, other one was log phase, other one was a stationary phase, and the last one was a stage of decline. This was basically a bacterial growth cycle. This is lag phase, this is log phase, this was the stationary phase, and finally, we are having it to be as a stage of decline, where the number of microbes gradually diminishes. Now, the organism generally is always preserved in a freeze dried form. The organism are generally preserved in the freeze dried form. So, you have to revive the organism, you have to make the organism rejuvenated. Rejuvenated means you have to make the organism active from the lag phase, and you have to put it up in the logarithmic phase. Now, you have to cross these barriers. Now, you have to put the organism somewhere in the logarithmic phase. This process is called sporulation, and we have to take the freeze dried microbes which are freeze dried, and then we have to prepare the first sporulation generation agar gland, where the nutrition, the content of the nitrogen will be lowered down, and as we go further, the nitrogen content needs to be gradually steadily increased. So, the first sporulation generation agar gland has to be prepared. It generally takes a period of 7 to 10 days. It generally takes a period of 7 to 10 days. After the first sporulation generation in an agar gland, we need to take the help of Erlenmeyer flask, where we have to keep the Erlenmeyer flask in a rotary shaker at 30 degrees Celsius. Continuous shaking will be there, and we will be going to the second sporulation generation. Now, I will be showing you a very small video where this video will show you how. This video will show you how gradually there is a growth of mold. Here the time is being demarcated. You see how the change in the complexion of this media changes. This is day number 3. Day number 3, the media is beginning to grow. This is day number 5 when it begins to appear. See this one. We have reached day number 8. So, at the end of day 8, the mold now is ready for production. Now, this seed culture is to be added to the fermentation tank. Sorry. Now, this seed culture is added to the fermentation tank along with the sterile fermentation media. So, separately you have to sterilize the media and then you have to aseptically convert or turn it into the fermentation tank, inoculate with the seed culture. So, automatically the culture will try to multiply, and when it multiplies it produces the product. Now, depending on the product, whether it is an intracellular one or an extracellular one, your process will change. That is what we are calling as separation, purification and recovery of the product. Let us see how it takes place. Now, the choice of recovery process depends on the nature of the product, the concentration of the product in the fermentation broth, the physical chemical properties of the desired product, what impurities are present in the fermentation broth, what is the intended use of the product. If it is of, if you require, and in fact intended use will tell you how much purify you have to make, minimum acceptable standard for purity, and finally the market price of the product. Now, if the degree of the enzyme purity is very high, automatically you need to be very cautious, very stringent method has to be used, and automatically it will raise to the increase in cost of the product. So, if this is extracellular, that is after production of the enzyme, that enzyme has come out of the bacterial cell. So, you need to take out the media as such. So, recovery process of extracellular products involve removal of microbial cells by filtration. So, first of all you have to remove the microbial cells, and then the broth is fractionated, extracted. See, extracellular means in the media, in the broth the enzymes is there. The broth is fractionated or extracted into major fractions using ultrafiltration, reverse osmosis, adsorption, liquid extraction, two-phase aqueous extraction, precipitation, and finally the product containing the fraction is purified by fractional precipitation, chromatographic techniques, and ultimately crystallized to produce the final product. Next, we are moving on. If the product is intracellular, that is the product is retained within the cell wall. If the product is retained within the cell wall, so you have to break the cell wall. Cell wall disruption is required. That has to be done anyway, one of the two methods. One is the physico-mechanical method, another one is the use of certain chemicals, which we are calling as the chemical method. Now, physico-mechanical method again can be classified into 4 broad types: liquid shear or a homogenizer. We can take the help of ultrasonication. We can take the help of breaking with a glass bead. We can take the help of solid shear, where we take minus 25 degree. So, by physical process we are breaking the cell wall, or sometimes we take the help of certain chemicals like detergents, osmotic shock, with the help of enzymes and with the phase of alkali treatment. This is a flow. Now, these chemicals are commonly used to remove or to bring out the enzymes which has been entrapped within the microbial cell, and after bringing it out you need to purify that particular enzymes. Similar is the case of penicillin. The same process you have to apply in most of the fermentation process. This is penicillinase, where the penicillin needs to be broken up. This is the flow chart showing the production of penicillinase enzyme. The same process you have to adopt, and ultimately we are coming to the end of the class today. Thank you very much.